So hi everyone, and thanks so much for coming to this presentation. This presentation focuses on the feeding ecology and behaviour of manta rays in Honeyfurrow Bay. My name's Hannah Moloney. I work for the Manta Trust and I'm a PhD candidate at the University of Sunshine Coast in Australia. Today what I'm presenting is a study by my co-supervisors led by Dr. Asia Haynes and as well as my PhD research, which is leading on from this. Reef manta rays are large planktivorous elasmobranchs and are classified as vulnerable to extinction on the IUCN Red List. They forage in tropical and subtropical environments and are generally low in nutrients. So Honeyfurrow Bay is the largest known aggregation site for reef manta rays worldwide, with upwards of 250 individual manta rays sighted at particular feeding events. Investigating the zooplankton dynamics and the food environment that leads to such feeding aggregations remains a key question for understanding their movement ecology. Currently faced with the challenges of a changing climate, any changes in their prey will ultimately affect the population, the movements, and the health of these ecologically and economically important megafauna. Manta rays are the basis of a multi-million-dollar ecotourism industry in the Maldives and worldwide. Zooplankton is tiny microscopic animals found throughout the world's oceans. Dense zooplankton is rare and patchy and is heavily influenced by environmental factors. So it's no wonder where zooplankton concentrates, so do the large animals, particularly the large planktivores such as manta rays and whale sharks. And by sampling zooplankton where manta rays are observed feeding, we can start to infer the relationships between the feeding dynamics of these animals. Dr. Haynes conducted the first study on feeding dynamics in Honeyfurrow Bay. From these results, they constructed generalized linear models to investigate the potential drivers of zooplankton biomass and manta ray abundance in the bay. In this first model, we can see the biomass was highest just following high tide, and manta rays were observed feeding when biomass was higher. The second model shows that manta rays were observed feeding, they were observed more commonly during feeding. This highlights that the bay is a really important foraging site for these animals. They also investigated the prey density threshold that can provide a benchmark for when the food environment is energetically rewarding for planktivores. To assess this threshold, Dr. Haynes constructed GLMs. They found that manta rays were significantly more likely to be observed feeding when zooplankton was in excess of 54 milligrams per meter cubed. This is the black dashed line. This is over twice the theoretical requirements previously calculated for metabolic rates for feeding. You can see here in the red dashed line. These are some dried dishes of zooplankton biomass, just to give you an idea of the magnitude of changes over the tidal cycle. So these samples were collected approximately 30 minutes apart. Zooplankton composition differed when manta rays were feeding compared to not feeding. The feeding samples were dominant in copepods, whereas the non-feeding samples were prominent with chitignae, salps, and eggs. Microscopy revealed that Undinula vulgaris was the most common species of zooplankton found in these samples. This suggests that there's offshore influence at this particular site. With research and answers to questions, always comes more questions. This is where my PhD comes into action. We plan to address questions leading on from the work of Dr. Haynes and Dr. Stevens. 1. Does the calorific and nutrient value of zooplankton influence manta ray feeding? And are there more mantas when there's more zooplankton? Do plankton dynamics influence feeding strategies employed by manta rays? Are environmental drivers the best predictors of manta ray visitation? How do manta rays spatially use Honeyfurrow Bay? Addressing these questions is important for understanding our understanding of prey availability, habitat use, and drivers of mass aggregation for a species that forages in nutrient-poor waters that are of relatively low productivity. In an attempt to answer these questions, we sampled Honeyfurrow Bay in the southwest monsoon in 2021 and will continue this sampling regime into 2022. The bay is probably more aptly described as a small inlet located just off the lagoon of Honeyfurrow Island. If we take a closer look, Honeyfurrow Bay is very small, so it's approximately 1 kilometre in length and a couple of hundred metres in width. So basically the size of a football field. And due to the high numbers of megafauna that's found at this site, it is protected as a UNESCO Biosphere Reserve, meaning that tourism activities are monitored by rangers and boating and scuba diving have been banned at this site. We plan to undertake comprehensive analysis using field methods by utilising a combination of drone technology, in-field, in-water sampling, underwater video, behavioural observations, and oceanographic data to assess foraging ecology, behaviour, and movements of reef manta rays and the environmental drivers behind these ephemeral hotspots. The zooplankton will then be taken back to the lab and processed so we can assess the biomass, community composition, and size structure of the prey of manta rays. When we take a closer look into manta grouping, we often see interesting interactions at feeding events. Mantas have been observed using 8 different feeding strategies, some group and some solo. They use these group strategies is believed to be linked to prey density. Mantas adopt coordinated or group feeding to improve prey capture, hydrodynamic efficiency, and avoid collision. Probably the most spectacular strategy of them all is cyclone feeding, which has only ever been seen in Honeyfurrow Bay and surrounding areas. Cyclone feeding occurs when zooplankton density is very high. The water can often be described as a plankton soup. Cyclone feeding is exactly what the name implies. Feeding chains basically loop into itself so the lead animal joins the trailing animal to create a circle. As more manta rays join, this builds up in the water column. So it can sometimes be 20 metres in diameter and reach all the way to the seabed. So the cyclone always turns anti-clockwise when viewed from above, and we think this is to do with lateralization. The manta's movement creates a vortex of water moving, which ultimately pulls the plankton in to where the filter feeding rays are consuming their zooplankton. This histogram displays preliminary results for this pilot study conducted in 2021. We have been investigating the reasons why manta rays employ particular feeding strategies and if this relates to zooplankton community and density. This suggests that there may be a relationship between higher zooplankton biomass and group feeding strategies such as chain and cyclone feeding. On average, there were more mantas counted during group feeding samples in comparison to the solo feeding samples. However, this is only early data and it requires further analysis. However, it has put us on a path of what to investigate further. We currently have a limited understanding about what drives zooplankton abundance into Honeyfurrow Bay, which ultimately leads the mass aggregation of manta rays in the southwest monsoon. This oceanography study, led by Plymouth University, will start in August 2022 and will go for approximately 12 months. Feeding aggregations have been shown to be linked to the tidal cycle, lunar phase, and wind speed, which generates strong currents that brings the nutrient-rich zooplankton water from the deep up to the surface and into the atoll. However, there's no robust explanation as to why this is the case. Also known is the influence of larger regional scale dynamics in modulating these oceanographic regimes. We plan to monitor the physical oceanographic conditions driving manta ray behaviour and zooplankton availability in and around Honeyfurrow Bay. Using an array of oceanography equipment, such as ADCPs, we hope to resolve the currents and changes in zooplankton density within and immediately outside of the bay to test the hypothesis of whether zooplankton accumulates within this site. We aim to estimate the density of zooplankton using the ADCP's echo sounder mode and to verify these results with zooplankton samples. So, in conclusion, zooplankton biomass and manta ray sightings in Honeyfurrow Bay is tidally influenced. Manta ray numbers peak inside the bay just after high tide, Honeyfurrow Bay offers prey density threshold in excess of what has been calculated to meet the manta ray's theoretical needs and far greater than observed at other planktonal aggregation sites worldwide in surface waters. The dominant copepod species suggests there's likely an oceanic origin and warrants further investigation into local hydrology and oceanography of Honeyfurrow Bay and Bar Atoll. This work has highlighted the importance of this site as a productive foraging region for large planktivores and provides support for ensuring adequate protection for this region. I would also like to briefly touch on some management implications that have arisen from this work. For example, this research has highlighted the importance of this location as a foraging site for large numbers of manta rays, suggesting the implementation of a code of conduct would be appropriate to ensure tourism activities do not negatively interfere with feeding activities. Also, to preserve this habitat and to stop further degradation of surrounding areas, the oceanographic conditions of the region should not be altered, i.e. no dredging or sand movement. Also, to preserve, and finally, climate change. Climate change poses an unknown risk to this aggregation site and to manta rays in the Maldives. It has been projected that in some tropical and subtropical regions, they may experience a decline of more than 50% of a decline in zooplankton. This would interfere with manta ray feeding. Continued monitoring of zooplankton will help to project large-scale changes in zooplankton biomass and a response to climate change in the Maldives. So with that, I'd like to thank you all for your time and listening into this presentation. And I'd also like to say thank you to the Manta Trust and Project Manta and all of our collaborating and funding partners for making this research possible. And thanks to the MMRI for hosting this event. It's wonderful. Thank you.